Uh, yeah, so well, welcome to the first Beckman graduate student seminar of the semester. I hope you're all staying warm and sort of thankful that you don't need to go to Beckman in person today to see some great talks. So today we got three speakers, Elizabeth Bello, Evan Anderson, and Suhnyoung Jun. So we're going to hear, their recent work on a variety of different topics. So I think we're going to start out with the first talk is going to be by Elizabeth. So Elizabeth is a second-year master's student in the entomology department. She's working under the direction of Dr. Marian Aline, and her research focuses on insect cuticle multifunctionality, and looking at brochosomes and understanding how biological surfaces can create new properties. So anyway, she received her associate's degree from Suffolk County Community College and then completed her bachelor's degree at Farmingdale State College. And she's a recipient of the 2021 Beckman Institute Graduate Fellowship. So with that, today Elizabeth is going to be talking about her work looking at brochosomes, so I'll let her take it away. So take it away, Elizabeth.
All righty, will do. Let me pull up my screen here. Thank you for the great introduction, by the way. Okie doke. Can everyone see that okay? Yes. All right.
Yes, looks good.
Okay, so hi everyone. As Michael said, I am Elizabeth Bello, and I'm currently working under the direction of Dr. Marian Aline in the entomology department. We focus mainly on bio-inspired materials and designs, and today I'm going to be talking to you about brochosome size variation and how that impacts the wettability of leafhopper wings. So to give you a little bit of background information, leafhoppers seen in these colored images here on the screen, they're sap-feeding insects that are primarily known as pests, but that's not really why we're interested in them. In our lab, we study insect cuticle multifunctionality. So we've looked at the nanopillar structures of cicadas and dragonflies, and then we've also looked at more hierarchical structures on fly wings. Leafhoppers, however, are unique in that it's not exactly their cuticle that gives them these this multifunctionality, but it's extracuticular particles known as brochosomes. And you can see those in the grayscale images here on the screen. So brochosomes are lipid protein granules ranging from about 200 nanometers to 1,000 nanometers in diameter. So they're really tiny. They're produced in the Malpighian tubules, an internal structure inside of the leafhoppers, and then they're excreted and applied through an anointing behavior onto the exterior of the insect. They're relatively understudied, but some of the hypothesized purposes include creating a moisture barrier to either keep liquids in or out, and also they could possibly be used as a predator defense and evasion tactic, as well as protection in some species, as they'll actually coat their egg masses with these brochosomes. So one thing I do want to mention is that the Beckman Fellowship really promotes interdisciplinary work, and we do a lot of that. So I've been working together with the Schroeder Lab. They focus on soft materials, and they've really been helping to characterize these brochosome structures. We also work with the Milkovich Lab, focusing on hydrophobicity and functionalities like that. And we also work with the Chen Lab, mainly on automated image analysis. As you'll see later on in my talk, I did a lot of manual measurements, and now we have the capability to do machine learning and automated image analysis, which makes the process a lot quicker. Before I dive right into my talk, I just wanted to show you guys some pictures of these really fantastic structures. These are all images of brochosomes at varying magnifications on the leafhopper wing. They're just really super cool, and I wanted to give you guys kind of an idea of what they can look like at different magnifications and how intricate these structures are. So starting the research, I chose these 8 different species that you see here on the right. They're from 5 different genera. So the 3 that are uncolored, those are from 3 separate genera, and then the 2 in blue are from the same genera, and the 3 in orange are from yet another genera. So getting started, I had 3 main questions in mind. I wanted to look at the brochosome size and how that varied from species to species. And I also wanted to look at whether or not the brochosome size varied from wing position to wing position. And I wanted to look at that question because I thought maybe as the leafhoppers were applying these brochosomes, the particles were being distributed either small to large or large to small, depending on the grooming behavior. So then the next thing that I wanted to look at was wettability and whether or not that varied from species to species or from wing position to wing position. And once I had those two ideas really nailed down, I wanted to go back and see if there was a correlation between brochosome size and wettability. So getting started, I collected local Illinois species, either using a sweep net technique or blacklight trap that you can see here on this left picture. And I identified them on site using Dimitriev's online interactive key. I would then store them in a -20-degree freezer and let them thaw out at room temperature for at least 48 hours before I examine them. To get a closer look at the brochosomes, I needed to use scanning electron microscopy, and I used the ESEM machine that we have at Beckman. And basically what I did is I took my 8 species, I had 3 specimens per species and 3 wing positions per specimen. And you can see that in this picture. At the top, we have our proximal wing position, which is all the way to the left, closest to the body, our medial wing position, followed by our distal wing position. I had a total of 72 images that I ended up analyzing. So I would take those images and I imported them into ImageJ Fiji, and I measured about 4,000 brochosomes in total. And the way that I did that, if you look at the top right part of this image here, I would take each brochosome and measure them at 3 different angles. So 0 degrees, 45, and 90 degrees. And the reason I did that is because they're not perfectly spherical, and that would kind of give us our best average diameter measurement. Moving into wettability, I used a microgoniometer, which is a piece of equipment that dispenses micro-sized droplets onto a surface, and it measures the contact angle as water is being applied to the surface, which is the advancing contact angle, and it also measures the angle as it's being removed from the surface, which is the receding contact angle, and in this case, it was done through evaporation. So you can see at the bottom here that our wettability or hydrophobicity is characterized by these angles. So if you have a contact angle that's less than 90 degrees, it's considered to be hydrophilic. If you have a contact angle that's greater than 90 degrees, it's hydrophobic. And if you have an angle that's greater than 150 degrees, it's super hydrophobic. And that's what we're after. And this part of the study was really inspired by these two studies that Rakatov and Gorb worked on previously in the early 2000s. So all the way on the left, this top left image, you can see they did an experiment where they had forewings and hind wings of leafhoppers that were both coated. Those are the white looking ones. And then leafhopper wings that were uncoated. So they removed the brochosomes and exposed the wings to their excrements. And you can see that the excrements really rolled right off those brochosome coated wings, but not so much the uncoated wings. So that was one inspiration for this project. And then the second part, down at the bottom, you could see that leafhoppers pre-molt are not hydrophobic, but once they've molted and applied these brochosomes, they become hydrophobic and super hydrophobic. And as far as data collection goes, I really did the same thing that I did with the SEM, same amount of specimens, and had a greater amount of measurements just because I had to do three measurements per wing position. So once I had all my brochosomes measured, I needed to determine really what was happening as far as variation is concerned. I did this using a two-way ANOVA, and I determined that wing position does not have any significant impact on brochosome size, but species does have an impact on brochosome size. The analysis also indicated that there might be something happening, an interaction effect happening between wing position and species, but I took a closer look at that using a follow-up Tukey analysis, and I was able to determine that it was more of a disorderly effect rather than it being a significant effect, and that was happening between the 3 erythronera species that you can see here on this graph, where there's a little bit more variation between the average brochosome size on different wing positions. So with this graph, one thing that I do want to point out, you can see that we have all of our species on the x-axis and we have our brochosome diameters. Each individual point is going to be an average brochosome measurement. So you can see that not only is there a lot of variation within one species, but there's also variation between different species, but not so much between those wing positions. So moving on to analyzing wettability, all of our species can be characterized as superhydrophobic. And you can see that in this blue horizontal region at the top, where all of our advancing contact angles are falling within this superhydrophobic region. There were a couple species that were in the hydrophobic region, but they were really like very near superhydrophobic. Their contact angles were just below that 150 threshold. Our receding contact angles were a little bit more inconsistent as far as individual measurements, but we believe that's because of a physics phenomenon known as Gibbs criteria, where as we're measuring the contact angle, the brochosomes are kind of moving along the wing surface and being pinned at different angles. So once I had both my brochosome size and my wettability, I wanted to go back and look at the correlation between these two. So what I did here is I created a graph where we have our average brochosome diameter on the x-axis and our average advancing contact angle on the y-axis. The region that's in green is going to be our hydrophobic region, and the region in blue is our superhydrophobic region. And I just overlaid a line of best fit on these data points. And while we didn't have any significance, it does appear that as brochosome diameter is increasing, our hydrophobicity is decreasing. And this can be explained by these two states that water can exist on a rough surface. So we have the Wenzel state, where the liquid is penetrating the solid surface and it's not leaving any air gaps. So it's considered to be either hydrophilic or lightly hydrophobic. And then we have this Cassie-Baxter state, where the roughness of the surface is actually creating an air gap between the liquid and the solid surface. And this is what we believe is happening in the case of these brochosomes. If we have larger brochosomes, there's going to be more air gaps in between them, and that water is going to be able to penetrate down into the cuticle. Whereas if we have smaller brochosomes, it's going to create this really tight impermeable barrier, and it's going to render our leafhopper wings super hydrophobic, as there's not going to be any room for that liquid to get down onto the cuticle surface. So in summary, I was able to answer all of those initial questions that I had. We learned that brochosomes vary in size both within an individual and also in between species. We also learned that it does not vary along a gradient on the wing, so there's really no distribution pattern happening there. In the same manner, we learned that wettability does vary between species, but it doesn't vary along a gradient on the wing. And it does look like there's some preliminary evidence of a negative correlation between brochosome size and hydrophobicity. And this is really important in creating new materials and new designs that are optimized for hydrophobic properties or other functionalities. So a couple future studies that I want to look at is determining the size of, determining the wettability of brochosomes separated by size. So in this experiment, all of the brochosomes were mixed on the wing. There's no separation of size, so they could be small, medium, large. However, if I were able to separate them, I would be able to really improve that correlation between brochosome size and wettability. The other thing that I'd like to look at is the wettability of leafhoppers with and without these brochosomal coatings, and that would give us a better idea of how much the hydrophobicity is improved by the brochosomes. And the last thing that I'd like to look at is the adhesive qualities, not only between the brochosomes, but between the brochosomes and the cuticle itself. And these are some images up at the top. I have looking at separating brochosomes by size, and then down at the bottom, I'm looking to do some AFM studies to look at the leafhopper wing topography and how our brochosomes might be interacting with that rough textured surface. So with that, I just want to thank all of my collaborators. Obviously, the Beckman Institute, I wouldn't be able to do a lot of this work without the equipment and resources at the Beckman. And we also have been in collaboration with multiple groups at the U of I, as well as the University of Texas at Austin and Northwestern University. So if you have any questions, I'd be happy to answer those at this time.
Great. Awesome. Yeah, nice talk, Elizabeth. Yeah, so we have time for a couple of questions. So feel free to just unmute yourself and ask away. Yeah, I got a question. So with the sort of your running hypothesis that the super hydrophobicity is due to the trapped air, and what was it, the Cassie, I can't read my own handwriting, Cassie Baxter model. Would that also be, would these surfaces then be, could they be omniphobic? And could you do the same measurements with, like mineral oil or something that's hydrophobic and see similar results, similar contact angles?
I believe so. There have been a couple studies, and I know that in the general realm of looking at wettability and hydrophobicity, some people choose to use different liquid materials. So in this case, we just used water, but I have also seen studies where people have used, like you said, oils and other things like that. I don't know what it would look like with oil, but it would probably be something similar to how it's repelling water.
Yeah, and I guess, do you have any sort of hypothesis for what's driving the size variability for the different grass leafhoppers?
Yeah,
Is it like a
so
geographic explanation, or what's leading to that?
it's kind of it's definitely still an unknown. We've looked at the overall body size of leafhoppers and the size of the bronchosomes that they're producing, and it doesn't look like there's any relationship between the size of the leafhopper and the bronchosome size. So we're not really sure like what's happening either internally to create that different size variation, or I mean, I guess it could be an external factor, but that is definitely one of our unknowns that we're looking into.
Mm-hmm. Cool. Yeah, so I mean, if nobody has any, I guess, last minute questions, let's thank Elizabeth again for her good talk, great talk. Cool. So our second speaker is going to be Suhnyoung Jun. So we'll give her time to pull up her presentation. So she's a fourth year PhD student in psychology, and she's in the cognitive neuroscience area and works with Professor Sepide Sarajiani in the Connect Lab. And her research is investigating the role of genetics on the I can't say that word, relevant features of the large-scale function connectome, mainly through fMRI approaches. So with that, I'll let Suhnyoung take it away.
Thank you. Thank you for your introduction. Hi, I'm Suhnyoung Jun, and I'm glad to share this exciting finding that dynamic trajectories of functional connectome are heritable and related to cognition. fMRI can measure brain dynamics as it acquires both signals from three-dimensional space and across time, resulting in four-dimensional data. From each of the nodes, which can be defined by your choice of brain atlas, we can obtain the time series or bold signal. Using the bold signals of every node, we can create static or time average functional connectivity architecture. Inter-individual variability in the human's time average functional connectivity architecture has been known to be subject-specific and predictive of cognitive abilities. However, such static architecture captures only part of the functionally significant properties of the functional connectome in human. In fact, the brain's functional connectivity architecture is inherently dynamic. It constantly reconfigures itself in an individual specific manner. This dynamic feature has been also known to contribute to inter-individual variability in cognitive abilities. Such association with behavior and cognition emphasizes the significance of dynamic characteristics of functional connectome states. So let's see what we already know from the literature. First, we know that behavioral and cognitive abilities are linked to connectome dynamics. We also know that some of the same behavioral and cognitive features linked to connectome dynamics are heritable. Therefore, there is a possibility that the behaviorally relevant connectome dynamics make themselves be heritable. However, there are multiple characteristics that can describe the connectome dynamics. So still, it is largely unknown which characteristics of dynamic connectome reconfigurations are subject to genetic effects and to what extent. To address this gap in the knowledge, our study had three main goals. First, we aim to answer which features of functional connectome states are heritable phenotypes. Second, we aim to investigate how much of the phenotypic variance can be explained by genetic effect using quantitative genetic modeling approach. Finally, we will examine how inter-individual variability in such dynamic features contributes to differences in behavioral performance across people. We use Human Connectome Project dataset and comprised 4 groups with different genetic similarity between subject pairs. This twin subject pool enabled the heritability analysis. And please note that the sex was matched between subjects pairs to control the effect of sex chromosome and other sex-related influences growing up. Additionally, we had 14 different cognitive measures and brain parcellation provided by the HCP team. Starting to the method part, the hidden Markov model is engineered to handle data which can be represented as sequence of observations and hidden states over time. So we use this hidden Markov model to extract 4 discrete brain states from the resting state fMRI. The 2 temporal dynamic parameters, which I will introduce in a few slides later, were also obtained based on the HMM inferred probability of state activation sequence. To address this first question, we obtained 4 behaviorally relevant multivariate connectome dynamic features, 2 temporal and 2 spatial. Specifically based on the probability of state activation map that you're seeing right now, were obtained from the HMM. And based on that, we chose the proportion of the total recording time spent in each connectome state or fractional occupancy, and the probability to transition between specific pairs of discrete states, or we call it that transition probability. By spatial features, we refer to patterns of edge strengths and ensuing topological characteristics of connectome states. Specifically, we chose time-varying functional connectivity between the default mode network and frontoparietal network, and also the time-varying modularity. To address the second question, we estimated the heritability of each of the connectome dynamic features with 2 different approaches. First, we use a 1 ANCOVA of the vector ceiling status and the similarity of each of the multivariate features. And then we quantified the extent of variance explained by genetics using the variance component genetic modeling. Finally, we addressed this last question. After identifying which features are heritable from the first and the second questions, we found their association with cognitive abilities using canonical correlation analysis. So let's dive into the results. First, we confirmed that the spontaneous connectivity time course can be described as a sequence of 4 whole brain connectome states. These states differ from each other in spatial organization, such as the strengths of functional connectivity between a default mode network here in frontoparietal network and global topology such as modularity. The proportion of time spent in each state was also different significantly, confirming that the 4 states obtained from the HMM are discretely different to each other. Here are the ANCOVA results that shows that if the subject pairs have more similar genetic makeup, they will also have more similar temporal dynamics of functional connectome. This will be helpful. Specifically, you can see on the left side that for a subject pair displaying similar state trajectories, they have very similar fractional occupancy and transition probability map. Which is not the case for the subject pair displaying the similar state trajectories on the right side. So the fractional occupancy pattern and the transition probability patterns are very distinct in here. However, that kind of genetic similarity corresponding to the similarity of the temporal features were not the case for the spatial features. The genetic similarity didn't really correspond with the similarity of each of the spatial features here. More interestingly, the heritability of these temporal connectome features were much stronger when they were treated as multivariate pattern compared to when their individual components were counted separately. Further, the effects were independent of head motion during the fMRI scan, global signal regression, or the chosen number of states, where we use 4 states here, and were absent in surrogate data set with preserved static cross-covariance structure. Using the genetic variance component models, we found that those temporal connecting features were best described by the ACE model. That is that 39% of the total phenotypic variance of fractional occupancy and 41% of the phenotypic variance of transition probability were explained by additive genetic model. Finally, we tested the association between the heritable temporal dynamic connecting features and the cognitive abilities, which were found to be heritable as well from the previous study. The C-state result shows that there's 2 significant associations here between the behavior and the heritable temporal dynamic connecting features. We tested the statistical robustness of the results by running 10,000 permutations. And as a result, we found 2 strong associations that temporal dynamic connecting features have, mostly with language and memory function, followed by cognitive control. Therefore, our study demonstrates that multivariate temporal features of dynamic functional connectome are heritable and are associated with cognitive abilities. Furthermore, we identified a best fitting genetic model for each dynamic connectome feature and provided an objective estimate of genetic and environmental contribution to each feature. Interestingly, the spatial characteristics of the brain states per se were not heritable nor associated with behavior. In conclusion, our work shows that the genetic makeup supports the individual specificity of connective dynamics and their contributions to cognitive abilities. So following up on this finding, we're now investigating the specific role of genetic polymorphism on the dynamic functional connectome. SNPs, or single nucleotide polymorphisms, are a type of polymorphism involving variation of a single base pair. More simply put, these are the places in the genome where people differ. Specifically, we're interested in the polymorphisms in neuromodulatory neurotransmitters involved in brain dynamics. Because the downstream effect of SNPs include changes in gene products and following biological changes, we're taking very strict hypothesis-driven approach to investigate the specific polymorphisms, which influences the transition probability and the fractional occupancy of connectome dynamics. I'd like to thank to Drs. Dadagiani and Altman, who gave me a great insight on the connectome dynamics and the genetic approaches, and Dr. Alderson and Dr. Deringer for their valuable input on theoretical understanding of statistical approach in genetic analysis. Thank you for listening. Any questions or, can be answered right now.
Yeah, great, thank you. Yeah, that was a great talk. We got time for some questions. So again, feel free to just unmute yourself and ask away. So I guess I can get the ball rolling. Like this is clearly not my field, and I feel like there's a lot to sort of understand. But what are the 4, what are the 4 states that that are sort of the transitions are going between? What do those mean exactly? And can there be more than 4 states?
So based on this hidden Markov model, we can extract more than 4 states. We can specify the model to obtain 4, 6, or even to 30 states if you want to. But we just put this random number, and we tested this number by trying different numbers like 6 states as well, and confirmed that the pattern of the genetic effect are still maintained if we use different number of states here. And we didn't really look into what each state mean. We really focused on their temporal characteristics of how the state's transitions are actually meaningful in how the brain dynamics work.
Okay. Cool. So I guess each state, yeah, I think I just need to think about that. But each state is like, I don't know, has a distinct,
Yeah,
is it like a spatial, a distinct spatial pattern to it?
Yeah.
Okay.
As you can see here, the four matrices, these are the state, the four straight states, and they have distinct patterns. We say a different network or different connecting states. And brains are fundamentally transitioning between each of the states we have in the brain. So by making transition from state one to state four and to state two like that, our brains are functioning to make decisions, wander around and think about something, looking into the visual stuff like that. But because our states are driven during the resting state fMRI scan, which is doing nothing and just thinking random stuff. So this is what the brains are intrinsically doing.
Cool.
Thank you.
Yeah, cool. Yeah, so I guess if there aren't any other questions, we can thank Suhnyoung for her interesting talk. Great. So our last speaker today is going to be Evan Anderson. So I'll give him an opportunity to pull up his screen. Hopefully he's able to. So Evan completed his undergraduate degree at the University of Illinois with a BS in psychology. He's currently a member of the neuroscience program and a graduate student in the Decision Neuroscience Lab. So his research investigates the neurobiological mechanisms underpinning human intelligence and decision-making using computational methods from cognitive neuroscience. So I should mention, Evan he's also a 2021 Beckman Graduate Student Fellow. So with that, I'll let Evan take it away.
Thank you for the introduction. Hi, yes, my name is Evan Anderson, and I'm here to talk briefly today about some research that we've done in the Decision Neuroscience Lab relating individual differences in human intelligence to dynamic brain network connectivity. We're studying intelligence in particular because the positive manifold, the shared variation among all cognitive abilities, explains a great majority of real world behavior, at least as you can measure it in a lab with a psychometric test. There are many theories of how the positive manifold can be decomposed into particular cognitive abilities, but essentially the take-home message is that individuals with higher intelligence, say individuals who will score better on a test of foreign language ability, will also score better on a test of mathematical ability. There's a positive the mutual relationship between cognitive performance across many domains. And traditionally, through much of the 20th century, this was thought of as the dominant model, which was used to conceptualize how the positive manifold, how intelligence can be decomposed into particular abilities, was what was called the Cattell-Horn-Carroll, which is illustrated here. But in the 21st century, so far, and probably through the remainder of the century, the majority of models that have been proposed to explain intelligence have been network-based in nature. And this dovetails with recent cognitive neuroscience research that in fact argues that literally network representations of intelligence are responsible for producing individual differences. One recent influential one is the network neuroscience theory, which says that intelligence, G, the positive manifold, depends on the dynamic reconfiguration of brain networks, modifying community structure, topology, and flexibility in service of sort of system-wide adaptivity. That intelligence and cognitive ability are global phenomenon that can be investigated using global approaches to cognitive neuroscience. One of the ideas that dovetails nicely with this perspective is what's called the criticality hypothesis, which says that the brain exists in something like a balanced state where it's easy to transition sort of in a dynamic way from moment to moment to any one state, that in the repertoire of state transitions, brain network transitions are generally easy to do. And that critical state represents a balance between what we call integration and segregation, ability to communicate with the ability to segregate into distinct communities. what we wanted to do here was try to investigate whether or not it was indeed the case that brain network dynamics and flexibility were able to predict intelligence. And to do this, we used a method called LIDA, leading eigenvector decomposition. It's understood and has been shown before that the brain transitions from segregated to integrated states in a global way. What this method allows us to do is actually view that process at a more granular level, where we can look at how individual networks transition from integration to segregation. We can discover those networks in a data-driven way. For a few reasons, this method has benefits over conventional approaches to dynamic brain network analysis. It's windowless, first of all. It's not clear how you what the ideal sort of sliding window to parse a functional scan into chunks is to try and extract intelligence. So we've used a windowless method that does not require you to select a window. It makes some assumptions, this method, about how the brain operates. Essentially, what it says is that brain network transitions, moment to moment changes in connectivity, are something like a Brownian process. That if you conceptualize, whether or not the brain state going from moment to moment is going to be integrated or segregated, if you were able to visualize that, it would take something like an energy landscape. And that when individuals enter brain states during, say, a resting state scan, when they spend time, for example, in a default mode state, that represents something like a minima in an energy landscape, and that as brain connectivity transitions from moment to moment in a resting way, the patterns of connectivity we observe will tend to sit in those local minima in this landscape. And another nice benefit of this approach is individual difference based. That is, we can look and say, here is a network we've discovered that predicts intelligence. Individuals who spend only 2 or 3 seconds in this network have lower intelligence. Individuals who spend 60 or 70 seconds in this network state, this phase state, they have a higher intelligence. Briefly, again, this is a data-driven approach. It does not require windows. It works on an individual TR. When we take an fMRI scan, we have, for each chunk of the brain, each sort of parcellated unit of gray matter, we have an amplitude of bold signal, which is just the oxygenation that occurs over the scan. How this technique works is it takes that oxygenation and then rotates it into the imaginary plane 90 degrees, and then treats it as if it was an attractor or an oscillator around some point. We then take the real valued component of that signal, that's what's displayed here in the bottom right. Essentially, we take the first eigenvector of each of these phase transitions, which you can just think of as a vector that says whether this particular region is pointing up or down and what its magnitude is. And we can then cluster those eigenvalues together to get a dynamic connectivity, a dynamic connectome. And what that tells us is just whether or not these brain regions in their dynamic phase are oscillating in sync or out of sync. And we can then apply clustering to that. Here's a schematic of what that looks like. Again, there's a bold signal. It oscillates in the real valued plane. We rotate it 90 degrees. We take its angle, and then we keep the real valued component of that angle, which is just the cosine. And we can then build dynamic connectomes out of those. So using this method, we wanted to answer several questions about how brain dynamics may be related to intelligence. One of those we want to know is it indeed true that dynamic connectivity and phase transitions are related to intelligence? If so, what are the particular brain regions that are most strongly related to intelligence? What patterns of phase coherence between which functional networks in the brain are entailed? And we also wanted to know, do individual differences in the probability of observing those particular states, do those predict intelligence? Can we produce an individual difference analysis here that tells us if we say, for example, scan a person and they spend 30% of their time in this particular functional state, will they have a higher or a lower intelligence? And we also finally want to investigate the actual duration of these phase states, not just the probability, but literally the number of consecutive seconds that someone dwells in a particular phase coherent state. Is that predictive of intelligence? And here again is a figure from one of the original papers on this method. showing a bold signal that's been rotated into this imaginary plane. And then we take the angle of it, and the cosine of that is what we're putting into this analysis. So to answer this question, we collected, as part of a larger study into studying interventions for modifying intelligence, we collected about 300 subjects worth of fMRI data alongside a large battery of cognitive tests. We processed that data using reproducible methods for removing motion and other nuisance signals. This was based on a package called fMRI Prep and then XCP Engine, using just standard conventional neuroimaging parameters. And then we parcellated those connectomes using an atlas called the Schaefer Atlas, which is hierarchical in nature. What it does, it takes existing sort of agreed upon functional network definitions, that is parcellating the brain into sort of large subunits that serve particular functions, and then subdivides them further into regions. We measured intelligence using a large and diverse cognitive battery of canonical tests of intelligence. We included a matrix region measure called figure series. We included a robust vocabulary measure. We also included the LawSat admissions, the law school admissions test analogical reasoning battery. We also included a measure of decision-making competence, which has been shown to be, if theoretically distinct, empirically indistinguishable, at least from measures of fluid and general intelligence. And using these tests, we constructed a latent representation of intelligence across individuals that accounted for, I don't know, it's like 65% of the variance across this entire battery using a single factor. And it was done using SEM modeling. So I want to briefly just show you some of the results of this empirical analysis. Again, it was exploratory in nature. We wanted to ask the question first, what brain states do people dwell in that are related to intelligence? And we found across a number of different clusterings solutions of brain states, which is how we're labeling these states from this raw empirical data. We found across multiple levels of clustering resolution that intelligence was significantly correlated with the total observed duration of a phase coherent state that integrated regions between the frontoparietal and default mode network. We did this analysis by Bonferroni correcting, so there were many other states that had non-significant relationships to intelligence, but here I'm only reporting results that survive that correction, and these are corrected p-values. Here are some graphics of what these integrated states that communicate between, in the orange, actually the red-orange default mode, and kind of the yellowy frontoparietal. These are states that we observed at several different levels of clustering resolution, and I've graphed here the probability of existing in that state, which is just a proportion of the total scan that someone sits there, against their normalized general intelligence. We see that for several levels of resolution, the most observed integrated state, again, is this pattern of multi-network connectivity. And also interestingly observed, and we see this in other parts of the study as well, that this gray state here at the top, which represents segregation, is inversely related to intelligence. What that tells us is that individuals who spend a longer duration or a greater portion of the scan in this sort of segregated, unsettled state where there's not a dominant pattern of dynamic connectivity that emerges, individuals in that state display lower general intelligence. And here's some more figures. We observe this same phenomenon as we increase the clustering resolution up to 18. The size of these maps shrinks because the clustering solution is identifying sort of additional states that are sort of subdivision of this larger pattern here that pull variance away. But we see sort of this same skeletal framework here, which looks a lot like the traditional task negative default mode network. And again, also includes regions from the frontoparietal network, and these kind of green regions here at the base are cortical limbic or extended default mode. So this tells us that indeed there is a relationship between the patterns of dynamic phase connectivity and intelligence in these individuals. Briefly, we also looked at some temporal states that for which we quantified the actual duration of connectivity, trying to just here demonstrate, how many seconds someone spends in a particular state consecutively. Does that relate to intelligence? We found some other interesting results there. This, for example, is what's called the task positive network, which is a canonically identified network that's present in many fMRI task-based studies. And we also observed, again briefly, that we also see the duration of a segregated state is important for predicting intelligence. Individuals who spend a longer duration in this unsettled state where there's not a dominant phase connectivity pattern display lower intelligence. There are several conclusions of this work. They suggest, I think, overall that dynamic resting bold signal coherence may be a predictive biomarker of individual differences in general intelligence. There's a distributed network of phase coherent activity across prefrontal, orbifrontal, and default mode regions that is associated with intelligence. And the attractiveness of this state during brain dynamics is highly correlated with intelligence. And we also observe, consistent with predictions made by various network neuroscience theories of intelligence, that individuals who display greater levels of whole brain segregation also display lower intelligence. So thank you for listening. I'd like to thank everyone in the Decision Neuroscience Lab and also everyone who was involved in this study and the PIs that collected this data originally.
Great. Yeah, thank you, Evan. That was a great talk. So we got time again for some questions. So again, if anybody in the audience has any questions, feel free to unmute themselves and ask away. I guess I can start it. So in these fMRI scans, that you're getting all of these this data from, are the people just sitting there? Are they actively doing stuff? So are you saying, like, in a resting state, if your brain, goes between these states, it's a sign of higher intelligence? Is that the way to think about it?
Yes, in a resting state paradigm, individuals are placed in a scanner. In our case, it was for, I think, 10 minutes or so, and told just to close their eyes and think of nothing or let their minds wander. We keep them from falling asleep, but yes, usually they sit there and think about whatever. The reason that one of the reasons that this is a useful paradigm for studying brain network connectivity is there's something of a hierarchical relationship between the sort of structural connectivity and organization in the brain, the connections, the white matter that span different regions, and the patterns of functional activity that will emerge at rest. Again, kind of, like I said, if you think of like an energy landscape, there are places, minima in that landscape that are more attractive. And that's an emergent phenomenon and is thought to be related to this sort of this multimodal sort of hierarchy that relates structural measures of connectivity to functional measures of connectivity.
Oh. Oh, great. Yeah, question.
I have a couple of questions. Great talk, Evan. So one question is whether you get this kind of coordination, this kind of coherence in task-related fMRI that's correlated with intelligence.
Yes, it's hard to do a task-based imaging study of general intelligence overall, I guess is the first thing I'd say.
No, but if you have a task and you look at coordination within that task, does that relate to measures of general intelligence? So you don't have to do it for an intelligence test per se, but if you do these, just any kind of a task, yeah.
Yes, with the caveat that I don't know that, I'm not sure that we've applied that this method LIDA has been applied to task-based data in particular.
Got it. Okay, that's okay.
But yes, in general, yes, and there's task-based studies of either fluid abilities, but many other abilities as well that have shown that you see, again, regional specificity, and many older task-based studies were done in a region by region way, not a network-based way. But yes, there is a relationship between the network connectivity you observe in tasks and, again, this same underlying sort of intrinsic dynamic connectivity that occurs at rest.
Okay, I think that answers it. And then another question I have is a more kind of future looking. So as you know, there's a wide-ranging and free-for-all literature and brain training. So if you were going to take this theory as motivating some suggestions, some policy or research directions in improving mental capacity, what would you suggest? What would be the most promising directions for brain training?
I think that the literature on transcranial stimulation and brain networks, it aligns with the findings we have here. And the findings of that literature is that it's possible to drive or potentiate greater network integration by having individuals exist in functional network states for longer durations. Now, and there's empirical work doing that with TDCS, stimulating the brain, and that's sort of I think that's a good framework for thinking about brain training and cognitive training in general. That the hope is that individuals who spend more time performing a task or acquiring skills that can transfer to unlearned domains, and one mechanism for that might be altering the connectivity of brain networks. And this work in particular suggests that it's not just the physical makeup or it's not just the spatial extent of a network. It's how prominent it is relative to other networks, and even it's the duration that individuals spend in it. And so, is it possible to design brain training paradigms that try to in particular target, say, the duration of a state, how long someone can maintain it for a while, or how easily someone can transition back into it? Like those would be, I think, the kind of principles that you could maybe take from this in trying to generate some sort of brain training program that was, again, based on trying to induce individuals to alter dynamic profiles of connectivity as opposed to just spatial extent. But again, with the idea that will hopefully be a mechanism for inducing transfer.
So could you talk about some behavioral ways to do that?
Well, in previous research that the lab has done, that Aaron's lab has done, and others on the Insight project, which was a large brain training program that used various modalities. That took the approach of using cognitive training games that were multimodal in nature, multiple games at a time, target abilities. And one of the other performers
but that didn't work, right? But that didn't work.
Kind of no. Well,
that's good to know, right?
That's a good way to put it. I think in general, that's the take-home message. But there were other performers who experimented with more diverse batteries, I guess is where I'm going with this. That included many, many more games, trying to directly, the thinking being, if you train people to do 20, 30, 40, 50, 60 different tasks, and there were that many tasks, you were, through whatever mechanism flexibility occurs in cognitive switching, you were training that directly by making them do it over and over again. And that sort of dynamic switching based perspective, I think that aligns with this research. And so again, it makes me think that the thing to do would be to find behavioral paradigms that induce that sort of switching in particular, that would hopefully potentiate this network dynamic in a way that would allow the, even without TDCS, potentiate some sort of plasticity in the brain that would make those transitions faster.
So would that be consistent with the recent findings that show that activity diversity is predictive of reduced cognitive declines with aging? It's like the more different things you do, which, I'm not sure that's really consistent, because I can imagine the more different things you do, if they're not coordinated, might work against that. So I'm just trying to think of how this might inform some clinical trial of putting people into different sorts of activities to.
Sure. So this Slider framework makes the assumption that the global pattern of dynamics that we observe in the brain is really an emergent result of multiple different subsystems that are all oscillating. So there's this big chaotic pattern that looks like transitions, but that there are underlying subunits that are important. Now, we don't know what those subunits are in this analysis. We just,
So I just want to interrupt and say I want to be cognizant of the time.
Sorry,
Perhaps,
I'm
perhaps
monopolizing,
this can
sorry.
spark a conversation offline, but I did want to, I guess, wrap up, give people time to move on to if they have obligations at 1. So yeah, so I wanted to thank our speakers today, Elizabeth, Suhnyoung, and Evan for their interesting talks. And the next Beckman seminar will be, I guess, in 4 weeks from now on March 2nd. So look out for that information to come. So thank you all again for coming, and thank you to our speakers.